So good morning and welcome to our Alaska Collaborative on Health and the Environment teleconference. This month's call is titled The Role of Mycoremediation in Alaska, Applications and Limitations of Mushroom-Based Bioremediation in Cool Climates. So we're delighted that we're joined today by our local Alaskan Fungi Alliance, their business advisor and owner of Paxaro Solutions, Jacqueline, a mushroom specialist, and one of the owners of Far North Fungi, Gabe, DeGange, and an environmental geochemist and the owner of Sustainable Earth Research, Dr. Birgit Hagedorn. So welcome to all of you guys, and thanks so much for joining us today. My name is
very much.
It's great to have you guys. My name's Nick Reardon, and on behalf of Alaska Community Action on Toxics, I'll be facilitating today's call. For those of you guys not familiar, CHE Alaska is a regional partnership group of the National Collaborative on Health and the Environment. And our aim is to advance knowledge and effective action to address the growing concerns about the links between human health and environmental factors. For more information, you can find on our website, akaction.org or healthandenvironment.org. On to business. I sent a link out with the presentation slides for this morning, along with the dial-up instructions. And so you can also find those, a link to the presentation slides if you go to our website at akaction.org. And just click on the title of today's call. It's the top of the feed there. As usual, this is an audio-only teleconference, so it's not a webinar. So when you get those slides, you'll just have to scroll or click through them yourself, along with the speakers. And so we've got an hour here, and so without further ado, welcome again to Jacqueline, Gabe, and Birgit, and we're delighted to have you guys on the call, and begin when ready.
Right. Thank you so much, Nick. Sorry about jumping in there for a minute. I got overexcited. So
you're good, Jacqueline.
All right. Well, good morning, everybody. This is, I know we're all getting very used to teleconferencing and using different technologies. I'm new to kind of trying to present this way, so bear with me just a little bit. If everybody has their slides, we will do our best to remember to tell you which slide we're on. So I'm on slide number 1. I'm going to do the welcome. I'm Jacqueline Summers. And after the welcome, I'll hand this presentation off to my colleagues, Gabe DeGange and Birgit Hagedorn. So welcome, everybody. We're very excited to be here today. And one thing that I'd like to start with is that this technology is actually something that's been established for quite a while. The new application that we're doing is using it specifically in Alaska. And so what we're hoping to do is give you an introduction today and then encouraging you to reach out to us. We'll have our contact information when you have specific questions about maybe an application in your area. So moving on to slide 2, we are the Fungi Alliance. And the outline of today's presentation is that I will give you a bit of a brief history and an overview of the actual business part of it, and then I'll hand it over to Gabe and to Birgit, and finally we'll wind up with some questions and answers. So that's on slide 2. Now I'm going to quickly move then to slide 3. So on slide 3, you'll see the contact information that I referenced before. There's 3 different businesses collaborating with the Fungi Alliance. There's my business, Paxaro Solutions. Then there's Far North Fungi, owned by Allison Dunbar and Gabe DeGange. And the last one is Sustainable Earth Research, owned by Dr. Birgit Hagedorn. So working together, we've created a social enterprise. We're a for-profit business that is working toward a social mission. So the Fungi Alliance is a social enterprise, for-profit collaboration, providing services, consulting, and products using fungi for environmental soil remediation. So as you can see, we're using a market-driven approach, but really have the idea of trying to do something to help improve the environment. So the Fungi Alliance actually came together 2 years ago. It's now been 2 years since we've been pursuing this idea, and we met kind of in 2 different ways. Birgit took a class that Gabe and Allison offered through their business, Far North Fungi, and Far North Fungi offers edible mushrooms. Many of you in Alaska may have seen them in Anchorage at the different farmers markets. And Birgit got excited about what the possibilities for using fungi for the environment could be. So she and I met separately at a class on how to launch new businesses. And we all came together subsequently and have been exploring this idea ever since. So who are the Fungi Alliance? The Fungi Alliance, moving on to the next slide, slide 4, we're 3 businesses, but 4 people. There's me, Jacqueline Summers, and then Allison and Gabe with Far North Fungi, and then Dr. Birgit Hagedorn with Sustainable Earth Research. And we each bring a different skill set to our collaboration. So I have a background in business consulting, and then Allison and Gabe are deeply knowledgeable about fungi and how to grow them and how to apply them. And that's a key piece of this business. And finally, Dr. Birgit Hagedorn has a background as an environmental geochemist, and her analysis and ability to specifically sample and apply this technology is a linchpin for what we're trying to do. Each place that we would want to apply this needs to be customized and analyzed, and she provides that experience. So with that brief introduction, I'm going to hand this off to Gabe, and thank you very much for your time today.
Hey everyone, my name is Gabriel DeGange, and I'll be talking a little bit about the fungi side of the mycoremediation. So in order to understand why mushrooms can be a powerful tool in remediation, we first kind of have to understand a little bit about their life cycle. And so if we all turn to the page 5 of the slides, you can see a diagram that kind of is an oversimplified version of a standard macrofungi or mushroom producing fungi. And it all starts with kind of what we're all familiar with the actual cap and stem of the mushroom, which will produce spores underneath the gills. And the spores are similar to a sperm or an egg in an animal. They're a gamete that will go on, and when it lands, it can germinate into a hyphae, which is essentially like the root or the body of a fungus, because the mushroom can really be considered mainly like the apple of the tree. Whereas unlike a tree, the fungal net or the actual body of the organism is underground and you can't really see it that often. So once the spores have germinated, if 2 of these hyphal nets meet one another, then they sexually reproduce. And at that point, you have a mature fungus that can go on to produce other mushrooms, although the actual production of these mushrooms depends on a lot of other environmental factors. So in a lot of cases, if we turn to slide 6, you can see kind of like a typical niche that a fungi might live in the wild. So these are known as mycorrhizal fungi, and it's thought that about 93 to 98% of plants actually pair with fungi in their root systems, and these fungi will trade micronutrients for sugars with the plant, and they'll help link different plants together. So a study in the late 90s in British Columbia actually showed that these trees will actually send their saplings sugars through the fungus, and the fungus will actually facilitate these sugar transfers, and in return they skim a little bit off the top, kind of like an interest rate of sorts. And the other thing that these mushrooms are really good at doing is providing metals and other essential non-organic nutrients and stuff to the trees. And this will be important later on. But to kind of get an idea for what fungus operate like in the wild, the largest organism on Earth can be seen on page 7, and it's actually a fungus, a parasitic fungus that kills trees. So it's been growing in Oregon for about 8,000 years, and it's about 2,400 acres in size. And so these fungal nets can be, extremely pervasive and large, and I think there's multiple 100-acre fungus nets that are nearby, and they essentially are surviving off of the trees, the organic molecules in the trees, and breaking those down for food. Obviously, in mycoremediation, we don't want to be unleashing a parasitic fungus, so the ones that we try and look at are actually more like scavengers than actual predators for the organic compounds. So on the next page, on slide 8, you can kind of see how the actual fungus will go about its life cycle and how it'll live. Pretty much these hyphae will spread out, and they almost move through the soil like worms, but they remain all connected. And so that's the reason that you'll actually see often what are known as fairy rings. You can find those where mushrooms will be growing in a small circle around, and that's actually them producing mushrooms at the edge of the mycelial mat. And so they probably started growing initially in the center. But what these hyphal nets will do is they excrete digestive enzymes similar to how we do in our stomachs. But unlike animals, what fungi do is they actually release these enzymes out into the environment. And then the hydrolysis reactions will break down nutrients, and the fungus will then absorb those micronutrients. So on page 9, you can see kind of a simplified version of these enzyme substrate complexes and how fungi will use those to break them down. And fungi are really the masters when it comes to just using enzymes to break stuff down, whereas animals are kind of fairly limited in terms of the types of molecules that we can digest and process. Fungi really depend on this for everything. It's their first line of defense, defending against predators or infections. Since they don't have 10 to 11 layers of skin protecting them, they really rely on being able to throw specific enzymes at whatever they're coming into contact with, whether that be a potential predator or it be some sort of food product. And so this is kind of what we humans can piggyback on that skill set. And if we turn to page 10, these are kind of the lignolytic enzymes are kind of the reason why we zeroed in on this particular group of fungi, which are known as the white rot fungi. But these lactase and peroxidase enzymes are really powerful, mainly because if you turn to page 11, white rot fungi have specifically evolved to degrade trees, and specifically the lignin molecule in trees, which is really what separates a tree from a blade of grass is the lignin molecule. And these molecules are huge. You can see the structure of it in the bottom right. And they also resemble a lot of the structures of certain contaminants that we find in kind of plaguing human society. So at this point, I'm going to turn things over to Dr. Birgit Hagedorn, and she will be able to discuss the chemistry side a little bit more.
Yeah, good morning, everybody. Thank you, Gabe. The ability of producing these enzymes really gives the mushrooms the ability to break out or break down contaminants, organic contaminants, and these enzymes are highly adaptive. That means they can face all kinds of different organic structures. If we turn to page 12, there are a number of persistent organic contaminants, and those contaminants have been tested with mycoremediation, mostly in laboratory or benchtop studies. A lot of tests were for petroleum hydrocarbons, gasoline, diesel, crude oil, polynuclear hydrocarbons, chlorinated compounds like PCBs, solvents, dioxins, dioxanes, and chlorinated pesticides, pharmaceuticals and personal care products, and bacterial contamination. So it's a vast amount of contaminants that have been tested in the laboratory and benchtop studies. And they all were quite successful, so demonstrating the potential of using mushrooms for mycoremediation. I want to specifically go a little bit deeper into petroleum hydrocarbon remediation, because that's what we focused on at first, and I think that are probably the most studies are for petroleum hydrocarbons. We'll also touch a little bit onto the chlorinated compounds. In terms of pharmaceuticals, personal care products, there are studies out there that just created inoculated bottles to treat runoff or wastewater filtration systems, where water is just filtrated through mycelium substrate, or bacterial contamination, just enhancing stormwater collection systems with myceliated straw balls or baffles is a way that is featured in the literature. Going back to petroleum hydrocarbons, this slide gives you just a little overview of what they are. Crude oil is the primary precursor of most of the petroleum hydrocarbons. And you see here, crude oil encompasses carbons from 2 carbons, or molecules from 2 carbons to 56 carbons. And then below that are distillates like gasoline, diesel, different kinds of fuels and oils. And the characteristic of these fuels and oils depends on the molecules, how large those molecules are, how many carbons they contain. Like gasoline is really fluid, asphalt is very viscous and stiff, depending a little bit on the temperature. What they have all in common is that they are not soluble in water, so they are mostly remaining, or they are remaining in the soil unless they are a free product, so they would just be on top of water if they make their way into the groundwater. Going on to slide 14, it's a little bit more about the structure of the petroleum hydrocarbons. So the molecules that are contained in these petroleum hydrocarbons can be chains, they can be cyclic, they can form aromatic rings like the toluene. Polycyclic aromatic hydrocarbons are aromatic rings that are connected to each other. There are over 100 different PAHs, and they are contained in gasoline and as well also in crude oil. And then crude oil, in addition to these structures, has also sulfur, nitrogen, oxygen, and metals. And it's probably the most toxic hydrocarbon because it contains also PAHs and all these other structures. Looking at those structures and comparing it to the lignin shows that the lignin is a similarity, and that's probably why white rot fungi are really destined to degrade these kind of molecules. There are a number of studies that show and demonstrate how successful white rot fungi can degrade these hydrocarbons. And I have outlined here a few, or named a few. At the end of this presentation, you can find a reference and this literature attached. So if you want to look that up and read about it in more detail. Basically what these people did, there are 2 different kinds. They either followed the degradation pathway, so they analyzed the initial concentration and the end concentration. Some of these studies also analyzed degradation products. Ideally, we want to see that all these petroleum hydrocarbons are actually forming or mineralizing, so they degrade into carbon dioxide and water. Of course, that's not normal always the case. So studying the degradation products is very important, and I will come back to that when we talk about the chlorinated compounds. Other studies did not analyze the concentration. They are more looking into germination successes. So if a contaminated soil, if the contamination is high enough, we won't see any vegetation progressing. And then they just look at which point a germination increases. For example, Sedu, he measured the germination of maize in contaminated soil. He found out that up to 15,000 ppm, the soil is recovering by applying oyster mushroom, and he found 100% germination of maize. For all of these studies with petroleum hydrocarbons, the oyster mushroom was the most successful for petroleum hydrocarbons, and that's also the oyster mushroom or the mushroom we are using in our pilot studies. Other compounds like chlorinated compounds, here you see the structure of polychlorinated biphenyls, PCBs. There are 209 different congeners. It's 2 aromatic rings that are connected, and they have a different amount and position of chlorine attached to these aromatic rings. There are studies with ligninolytic enzymes, so enzymes also from white rod mushrooms, and all these studies pointing out that the oyster mushroom is maybe the most useful and successful in degrading these compounds. And these enzymes are splitting the aromatic rings, and they are dechlorinating the aromatic rings. Other mushrooms have been studied as well, and those don't produce ligninolytic enzymes. They belong to the Ascomyzota fungi like penicillium, and people found that they are dechlorinating, so they don't really split the molecule apart, but they are dechlorinating. Again, it's important here with these compounds to follow through the degradation products and making sure that we don't produce more harmful compounds instead of what we started out with. Other chlorinated compounds here, this is a solvent. So while PCBs are not very soluble and don't really leave the soil, they are just in the soil. The trichloroethylene is a product derived from industrial cleaner, and it's highly soluble and found in groundwater. Again, there were studies about fungi, and people found out that fungi can degrade or change or transform the solvent into a less harmful product like trichloroethanol by breaking up the double binding and attaching an OH group to the molecule. Looking to the degradation from trichloroethylene to ethylene, there can be byproducts like vinyl chloride, which is even more toxic than trichloroethylene. So again, it's very important when we start observing or start doing these studies to make sure we understand the degradation products as well. Looking at these chlorinated solvents, they are soluble in water. That means we find them in the groundwater. And there's a limit to in-situ application of fungi because they only grow so far and they do not really grow in water. There are tests that applied mycelium filters, so just filtering the groundwater through myceliated substrate. Another way would be to inoculate trees or tree roots with mycorrhiza, and Doty and others published a paper about that. So they found a mycorrhiza and endophyte that can degrade trichloroethylene, and they attached those to poplar, and planting the poplar, having the root system going down into the groundwater, they were able to degrades TCE almost completely. So this is actually a study, and this is a method that is currently applied in real remediation projects. So going away from the organic contaminants, there's also a way to remediate metal contamination in soil. Mushrooms are known to sequestrate metals, so to accumulate them in their bodies and their stripes. But there's also maybe a cheaper way to create filtration systems with mushroom material, and that can be everything from dried mycelia, live mycelia, spent mushroom substrates, or mushroom substrate that is immobilized in calcium alginate. And so all of those studies exist. The processes are, there are activated ions in the mushrooms and on surfaces of mushrooms, so you can have ion exchange, chemisorption, chelation, which just immobilize and absorb the metals. Studies say that there's in between the pH 5 to 6, it's most favorable for most metals, but so it depends on what metal you are looking at, which pH you want to apply. Yeah, so that's what I have for literature studies and how mushroom and fungi can be applied in remediation. Again, there's a lot of lab studies, laboratory studies, and there's a lot of benchtop studies out there, but the real world application is really the challenge. And so those challenges are finding a method that we can apply to create enough enzymes to treat the soil, containing the contamination as long as it takes to treat the soil, and finding out what the degradation product is. And so I want to give the microphone back to Gabe. He will talk a little bit about our first pilot study here in Alaska, which we performed with the oyster mushroom, which is actually cold tolerant and seems to be the most versatile for all these contaminations. We got this study, or the plot is at the Anchorage International Airport and Menzies Aviation, who takes care of the contaminated soil through refueling of airplanes, was providing us with the plot and supported our study. So a big thank you to Menzies.
Thanks, Birgit. So if we all turn to page 18, you can see just a picture of our small pilot study that we did, and kind of the work that went into this before we actually got to the site was we had to grow out the fungus in a volume that would actually be able to go into the contaminated soil, with the idea of kind of a 1 part inoculated organic material with the fungus, and 4 parts contaminated soil was kind of the final volume that we were looking to achieve. And so we did 2 separate batches of that. We did, one batch was spawn, which is a grain that has the fungus coated in it, which gives it a nice food source, as well as each grain, when you break it up, will be covered in the fungus, and you can spread that out kind of homogeneously through the soil. And that's the way that we actually do for commercial food production. That's what you do into sawdust, which is kind of your main food source. So what we did was we essentially did that for part of it, where we took the grain inoculated, we mixed in some undigested sawdust to kind of give it some food to chase after and a nice lignin-rich substrate, so that it would have to produce a lot of those enzymes that we showed earlier. And that was kind of the intention with part of it. And the other part was to see if we could get away with using spent substrate. So sawdust that had already produced commercial quantities of food production and see whether that putting in kind of a waste product from mushroom farms would actually serve as well. And so kind of what we figured out was that the younger material, the spawn with the grain, was actually the way to go. Just because the mushrooms age and they're not nearly as viable or as responsive, especially when you take something like a oyster mushroom, which in the wild would just be growing on lignin-rich wood products in the forest, taking them out and putting them in an open, exposed field with really low humidity. Definitely you start running into a lot of challenges that I think have really plagued mycoremediation since the 80s, which is kind of how do you take something out of the benchtop scale and bring it out into the real world with all the variables that you find and be successful. And I think a lot of the kind of failures to achieve larger scale success with remediation outside the lab have been due to just the difficulties of growing fungus. So if we turn to page 19, you can start to see at about a month and a half after we inoculated the material, you can actually even see the mushrooms poking out of the bottom of this main plot that we have. And if you look, the bottom part of it that's just like a visqueen plastic sheeting, and above that is weed guard. And the weed guard turned out to be a much better solution. While we did see some growth and remediation, like the fungus was clearly growing in under the plastic sheeting, and there are mushrooms you can see poking out there that look pretty large and happy. Overall, I think that CO2 levels were far too high underneath the plastic sheeting, and I think that the humidity was a problem. So if you go down to the 100 days, the next slide, slide 20, you can really see how the plastic sheeting inhibited the growth of the fungus, and which in turn, by day 100, you can see that on the weed guard side, the mushrooms have produced nice, big, healthy caps, and you're actually starting to see some primary succession. So you're seeing a lot of algae and other plants, as well as some vascular plants. In the bottom right of the left side of the plot, you can see that there's even like a little dandelion starting to grow, and there was also some grass growing. And the part that says weed guard in the top right of slide 20, you can see that's not frost or anything in October. We had a pretty warm October. That's actually the mycelium growing. So those are all spores that have germinated, and the mycelium mat is starting to knit together before rushing underground, as it is prone to do. But for us, that was really exciting because in this pilot study, we did not have time to initially train the fungus on petroleum hydrocarbons in the lab portion. So essentially, we were taking what is commercial mushroom spawn that hasn't been trained to grow on, diesel range organics. And so its first real introduction to oil was in the ground here. So that's really throwing a lot of variables at it at once. And so we were really pleased to see that the mushrooms had fruited out and produced spores, because once those spores hit the ground and meet one another, then you're going to have that sexual reproduction that we talked about on slide 5. And at that point, then you really start to see the fungus zeroing in on traits that are useful to either breaking down, diesel range organics or just surviving in this new environment in general. And that's kind of the exact type thing you want to see. And as I mentioned before, like ideally you would actually familiarize the fungus with your contaminant from an early stage. And there's been some literature that seems to imply that you can actually steer the fungus towards using enzymes specifically that will break down those hydrocarbons really effectively, to the point that you can actually eliminate most of the other food sources. So that's something that we'd really like to try in the future, is really getting in on the lab stuff before the ramp up into a bulk substrate that will then go into the contaminated site. If we go on to 21, this is the, so it's 9 days afterwards, and you can see in the bottom left kind of how the control looks comparatively, just as kind of a qualitative observation that on the side that had the fungus added, there's really a lot more going on, so you can really tell that it wasn't just the fact that there was weed guard on there and humidity added in, that those variables, that's not enough to show it. And those vascular plants showing up is a really good indicator generally of healthy soil, and that the environment is starting to recover. So at this point, I'll turn it back over to Birgit to talk a little bit about the chemical analysis that we did.
Thank you, Gabe. So, just one more thing. So we use oyster mushroom here, the blue oyster, and Gabe and Allison use actually the same blue oyster, and you can buy it from them as a gourmet mushroom. So it's really very versatile. Going on to page 22. So we not only had the visual observation of growth of food bodies, but we also collected some data. So on the left side, that are data collected from Menzies. They have to collect data to find the initial contamination. It was a relatively strong spread of data we got from them, everything between 200 and 890 ppm in that specific area. So that was our initial contamination or contaminant concentration of these range organics. After the one summer, so July, August, and September, 3 months, 100 days, we collected sample and analyzed them, and you can see that the second set of analysis concentrations, and all the concentrations were down between 120 and 150 ppm. We left the site installed over winter, and it was a warm winter, 2018, 2019, so we expected that something more will probably happen, and we started sampling again beginning of June, and there was even again a little more degradation going on over winter. So slightly lower concentrations, all around 50 ppm. So way below the regulatory limit. This summer was really hot, right, and warm. So we didn't really had a lot of mushroom growth. So we learned that to maintain a site, if it's really extreme, it's important to monitor and keep the moisture up. But on the other hand, the concentration was already down. We sampled again this October, and sent those samples actually to a commercial lab to just confirm our analysis, and so found the same concentration, so 40 to 50 ppm. So what did we learn? If you move on to page 23, based off our experiences, we created a process flow, how we would go if somebody would give us a contaminated site, or we want to treat and remediate a contaminated site. So the first step would be characterizing the soil, see if there's maybe the need of additional fertilizer or to adjust pHs. So we would just sample the soil, analyze the soil, and start cultivating a strain for this specific contaminant. We will still stick with petroleum hydrocarbons, and we would train in the laboratory the strain to the petroleum hydrocarbons. Then there's a point where we need to cultivate and extend the mycelium. So we would grow the mycelium from the petri dish out into a grain bag, and then use these grain bags to inoculate wood chips. And that is about 2 months. So that's the time that needs to be addressed when the project is planned. After that, there's a site preparation. So going on site and mixing in the wood chips. 1 part of wood chips to 4 parts of soil seems to be working well in terms of giving the mycelium enough food to create enzymes and to grow out in the soil, and then maintaining that over 1 or 2 years. And then after that, it's just monitoring the contaminant, monitoring the moisture seems to be very important. So that would be, if you have a contaminated site, that would be the process you need to follow in order to inoculate. We came up with kind of a fungi biopile, so it would be easy to upgrade a stockpile. We think it would, we will have berms that contain the contaminated soil. It should be lined with a plastic liner, have a little grading so we have a runoff if there is water going through the stockpile or the biopile. To collect contaminated water, it can be just re-injected on top or into the stockpile. And then we would just inoculate this contaminated soil with wood chips. We went further on and also tried out cardboard. Our vision was always creating a process and a method that's also applicable in remote and rural areas where we don't have a lot of access, so where transportation becomes really difficult. So going from wood chips to cardboard, one thing is we can recycle, so it's maybe better, it will be better applicable to areas where we don't have wood, and it's lighter, so it's not as expensive to transport. We also started experimenting with crude oil, and this year is really testing the strain, which strain would work with crude oil, which mushroom, and then also at the same time conditioning it. And at the top you see the blue oyster, on the left it's a control, on the right is the mycelium going out on 3000 ppm of crude oil. You can see the mycelium doesn't really grow that fast and that well, but it grows for the blue oyster. If you go down to the right corner, it's like page 25. The Stropharia doesn't even grow at all in crude oil, so it's maybe not a good candidate to apply in crude oil contaminated soils. To make our application more sustainable, so we tried waste materials, the cardboard. We also tried choke cherry chips, and that worked very well. You see going out the mycelium after 4 weeks, this white stuff on the cardboard is actually the mycelium going into the cardboard and the choke cherry chips. And then 8 weeks later, on the left bottom, you can see that the mushrooms are already going out of the mycelium. The cardboard had another advantage. It's not only cheaper and lighter, it also was faster. So the mycelium grew on average 2 weeks faster than compared to wood chips. We see this as a very viable method here in Alaska. It has minimal maintenance. You saw our experiment with sheeting, with plastic sheeting. We did not observe growth of mushroom bodies underneath, but the mycelium was still growing. So if you want to convert a stockpile, you want to avoid rain infiltrating, I think there is a way to just keep a plastic on top of it, because the mycelium can grow in the dark. It doesn't need light. It does need some air exchange, so a passive aeration tubing would be beneficial. Yeah, so what is next? We know there are many positive studies for many contaminants, so we want to keep going, testing out different mushroom strains, maybe going to native species, and also targeting other contaminants. PCBs would be on my first list. We really want to establish a business, and we don't want to operate as a citizen science group, which I like, but I think to really bring this to Alaska, we need to establish a business. We are working on really writing a white paper targeting all these agency regulations and make sure we comply with those, and we gather data that support the application of mycoremediation. And then we are looking for pilot studies or real applications or remediation projects. So that's the end of our presentation, and we welcome questions at this point. Thank you.
Yeah, thanks so much, you guys. That was that was wonderful, dealing with some complex topics in a pretty accessible way. We definitely have some time here, 15 minutes for some questions. So yeah, I'll invite those from our listeners. A reminder, just press *2 to unmute your line, and just state your name or affiliation, and fire away.
Hi, this is
Good
Patty.
morning.
My question is going back to the part where you were using the mushrooms to get metals out of the soil. So I guess my assumption is that the metals go into the mycelium and stay there. And so what do you do to get if the object is to get the contamination out of the soil, then you have to get the mycelium out of it. And what do you do? How do you do that? And what do you do with it once you separate it from the soil?
Hey, thanks for the question. Yeah. So one thing that has been kind of interesting is that with the metal accumulation is a lot of times what they find is the mycelium will actually transfer the majority of the metals that end up in the mycelium will be channeled up into the actual cap of the mushroom. And the kind of biological reason for that is they think that they're actually trying to remove that from the soil. Since fungi can't really move that well, they don't have the luxury that animals do of just kind of picking up and moving away from a potentially harmful environment. So they think that they're actually trying to get the mushrooms to be eaten by some animal who will then transfer them kind of away from that geographic area. So the difficulty then in terms of using fungi for remediating metals is you have to pretty much be monitoring the site and collecting those caps, as when they're produced, and collect them, and then you would have you'd have to dispose of them in a way. It would be a lot cheaper to move a small amount of dried mushrooms than it would be to do, to dig up all the soil and stuff. So it still could be a potential useful way to do it, but there are a lot more steps and barriers to that being kind of commercially viable, just because unfortunately it's not as easy as it is with the petroleum hydrocarbons to just kind of let them break them down. There is that kind of added step of having to collect the actual mushrooms. So yeah, you're totally right that it's not as easy with the metals for sure.
So I just adding to that, what Gabe said, just in a kind of more how to deal with the contamination. So the mushroom is actually taking it out. If you do live mushrooms, right, there are these applications where you just create an absorbent. But if you do live mushrooms, harvesting mushrooms and drying them would be a very lightweight disposal of metals. If you think here, Alaska, and having to transport it over far distances, and I think that's a big advantage in compared to like phytoremediation. So it's easy to harvest fruit bodies, and that pretty much means that you take everything away that is contaminated with metals.
Great presentation. My name is Vai Wari, and I'm from Sivokak, also known as St. Lawrence Island. We have two former Air Force bases that closed during the Cold War, and we've had community-based partnership project with ACAT since 2000, where we've identified PCBs, pesticides, and heavy metals. We're just below the Arctic Circle, and this sounds very exciting and a great way to see if a pilot study would work on our island. And have you looked, done any, use this method at formerly used defense sites?
No. The thing is, nobody is doing mycoremediation in Alaska. It's not, there is quite some applications in the lower 48. It's not here yet. So it's a brand new application. It's not brand new in terms of developing this, but bringing it to Alaska and making it work here. We would welcome a pilot study. So we should stay in contact and see if we can work something out. Thank you.
Thank you.
Hi, everybody.
If somebody else wants to jump in. Hi there.
Hi, my name is Ashley. Can you guys hear me okay?
Yeah.
Yeah.
All right, perfect. My name is Ashley. I'm at the University of Washington in Seattle in the College of Built Environment, and I'm really appreciative of your conversation and presentation today. Some questions that I think I might have missed while I was following the chemistry portion is when you were doing your pilot project at the Anchorage airport, how many samples did you collect from the site, and what was the size of the site? And then the other part is like, for a pilot study, talking about the person who maybe just presented one maybe in the Lawrence Island area, like what would be a budget for something like this that you're like would need in order to scale or to do effectively? I wasn't able to really follow that.
Perfect question. Thank you so much. So the first question to the pilot study here at the airport, it was a small plot because we just jumped on it. We barely just met, and then we got this opportunity and we didn't want to pass it off. So it was not, we had a total of 12 different plots. We changed all kinds of treatment. And so each plot was only 1.5 feet by 1.5 feet. For sampling, we collected 3 random samples per each plot and homogenized it. And we had replicates, and we analyzed every single plot and the replicate. And you can see the analyses are really tied together. So there was not a big difference from comparing the treatments. It was more visual than really in terms of how the contamination decreased. So it was an ad hoc project. It gave us a lot of insight and knowledge how to do this. The other thing, coming up with a good price, it really depends a little bit on the situation. So it depends on how big is the contamination. Is there already, can the soil, is it contained, or is it just really on the landscape? What would transportation options, because of transporting the material to the island, sometimes we can team up with somebody on the island or on the location to help growing out the mushroom maybe or mycelium right on site instead of shipping everything out. So there are lots and lots of variables. So coming up with a good price, we try to stay in the same price as other remediation, in-situ remediation would be, like just having a stockpile and maybe adding some microbes or some fertilizer and let it sit as long as it takes. We know that doesn't really work very well with PCB, so enhancing that with fungi would be very beneficial. But I can't really just give you I can't. So it would take some time to figure out what really the price would be. I don't know, maybe somebody at Gabe or Jacqueline want to chip in there too.
So we were talking to a company that had a fairly large, I think, 1,000 cubic yard stockpile, and just getting the wood chips and things that we would need, like just the barge alone was like $60,000 just to get material out there. So in a lot of cases, that kind of, yeah, like Birgit was saying, it just depends on the site and how much of it can be done kind of located here in Anchorage, because the goal obviously is to ship out as little as possible. So maybe for a site like St. Lawrence, given how remote it is, we would probably want to maybe have them stockpile cardboard or something in advance. And then we would probably try, since it is, there is a community out there and they do have some resources that we could probably use in terms of, space and equipment to do it. That would probably be a case where we would be looking to do as much of it out there as possible. And whereas this other site that we were talking about, this 1,000 cubic yard, that was just a remote, I think it was some sort of drilling related site that didn't have any communities nearby. And so in that case, we would have had to pretty much do everything in advance and then ship out finished materials. So it really definitely depends. And I think this is another reason why mycoremediation has had a tough time getting from kind of the benchtop into the real world, just because each site is really different, just not only with the makeup of the soil and how to grow the fungus, but just the logistics and finances in terms of just figuring out how to make a viable option out of the mycoremediation.
Yeah. Having said that, maybe just a final sentence. So if the community buys in, I think there are plenty of options to do that in a reasonable way, but it would need help from the community, from the people that are on site, helping to make it happening. Okay, thank you. So.
Sorry, and this is Jacqueline, jumping in right at the end, just saying that we've added grant writing services onto our business, so that if there were communities that wanted to access EPA funds or IGAP funds or something, we could assist in that way. So thank you very much for the question. We're happy to answer any follow-up questions by email. Again, our contacts are at the beginning of the presentation. So thank you, Nick.
Yeah, I'm just looking at the time here. I think we might call it there. And yeah, Jacqueline's point is excellent, that there's they're welcoming questions, guys. So pepper them in the future. I think this is a great. We're somewhere in the midst of a conversation. I'm looking forward to continuing. So yeah, thank you three so much for participating today, and everyone else who called in. I'll send out an email in the next day or two with a recording of the call and some details. Our next call will be at the end of April on the 29th, and our topic then is to discuss a recent lawsuit to the EPA to encourage them to regulate the use of oil dispersants like those used in the Exxon Valdez spill and more recent Deepwater Horizon. And we'll have on Claudia Polsky from the UC Berkeley Law Clinic and one of the plaintiffs in that suit, Rosemary Asheangrak from New Mexico, Alaska. So yeah, finally, I hope you guys would consider making a donation to ACAT here and to support our monthly Che calls. And yeah, your contribution helps provide the support we need to continue bringing you these calls each and every month. And you can give at our website again at www.aaction.org. And if you have any other questions or comments, feel free to contact us here at our office, 907-222-7714. Thanks again to everyone, and I really hope you have a wonderful rest of your day.
Thank you so much.